board on computer. So I just wanted to go over some notes, just go over the chapters for you guys. And then if y'all have any questions, just interrupt me or save them to the end or whatever, however you feel like doing it. Chapter 4 is all over just the cells. So my 7th grader right now is learning cells, and you would have learned it probably also in Bio 1 when you were probably a freshman in high school. So hopefully this is review for you, and it'll be like the easiest quiz or easiest exam that you go through. I'm not going to pull it up on the, hopefully this is big enough for you to see is what I'm trying to say, because I'm not going to make it big screen so I can kind of see what else is coming up behind these slides. So the gist of chapter 4 is that you want to know all the organelles and their functions. And then the intro is a little bit about microscopes and different types of microscopes. So cells are teeny, teeny, tiny things. They are the simplest collection of matter that's alive. So they're the smallest unit that contains all of life's functions. So that's a really important theme that you'll hear throughout biology. And I don't know what other chapter, but I wouldn't be surprised if a question like that, it will be found on your final exam. Final exam, by the way, is a departmental exam. So it's different than the exams that I put together, but I don't have a lot of control over that. But I do know that is a big theme in biology that will probably be on that final exam, that cells are the smallest unit of life that still contains all the functions of life. So section 1 in chapter 4 is really just about microscopes. So it's just kind of memorization. Light microscopes are what you guys would use if you were in person in lab. Of course, we're not doing in-person lab this semester, but basically it just bends light, magnifies the image. Cells are generally too small to see much. With even a light microscope, we can't see a whole lot that's inside the cell, but you can see the outer parts of a cell. So you would have in lab done like a skin scraping, like taken a toothpick and scraped your inside of your cheek and looked for little skin cells on a slide, or taken, if you've ever peeled an onion, the thinnest part that you can peel away, not real thick, but just like the little tiny thinner than paper piece, is actually a solid line of cells, or the thickness of a bunch of cells. So you could have done that too and seen the difference between animal and plant cells, but you wouldn't see a whole lot other than the outline of the cell and then maybe the nucleus on the inside. So on light microscopes, I would know the 3 parameters of microscopy. It magnifies, resolution gives you your clarity, and then contrast is the difference between light and dark. This is probably stuff that Ms. Guevara covered maybe in the beginning of the semester for lab also. So light microscopes only magnify about 1,000 times. And then we can't see the organelles, like I said. So you can see in this figure, this is a good figure to just kind of give you an idea of how well we can see with our eyes, how much a light microscope gets us. And then we have something called an electron microscope that gets us even teenier, tinier things. So we don't have an electron microscope. You might, if you were going to like a bigger university like A&M or UT, maybe if you took a chemistry class or microbiology, you might have access to an electron microscope, but most of the labs that you would take would just be a light microscope. So electron microscopes are used to study the organelles. Then there's scanning electron microscopes that just shoot a beam of electrons and they make a 3-dimensional thing. And a light microscope is not really 3-dimensional. It's kind of just a 2-dimensional type image. And then transmission electron microscopes shine through it. So you can study really the inside of the structure of the cells. So I would just know the 2 types of electron microscopes and the difference between the 2. And then there have been some, science is always changing, so there have been some advances in light microscopes. And so we can get things pretty tiny. I think this is the slide to look at. And it's like, somehow they're freezing these things, but I don't think I'm not gonna really ask you about this cryo-electron stuff. Cell fractionation is where we break up the cells. So if you know what a centrifuge is, it's a little machine that you take vials of something and stick it in there, and it just spins it so hard so that it separates things by size. So the heaviest stuff will fall to the bottom, and you end up getting layers based on size and weight. Kind of like when you let, I don't know, olive oil and water, or olive oil and vinegar sit. Eventually it settles out. It's the same thing, but we force it to do that by spinning. So I would just know the term of cell fractionation. Once we've separated everything out, then we can take things knowing about what their size is and put that on a microscope and study what it is, or look at its characteristics. So on to actual cell stuff. The microscopes are how we see the cell, but what we're seeing when we're looking through the microscope is a number of different things. So I would know the difference between eukaryotic and prokaryotic cells, and that was in chapter 1, so it's kind of review, hopefully. I would know the difference between eukaryotic and prokaryotic cells. Prokaryotic cells, of course, are like bacteria and archaea, really, really simple, old organisms, teeny tiny, like one cell is what makes the whole animal. Whereas the more advanced things are eukaryotic cells. So I would know what the difference is between the two of them, but also what they share. And the things they have in common is they all have a plasma membrane that encompasses them. They have something inside of that, inside the plasma membrane. There's some kind of a fluid. So just like we've got blood and fat inside of us that kind of cushions our organs, and our organs don't just like all fall down to the, bottom of our pelvis. They sit floating. Same thing with a cell. There are things inside. There's a matrix, the cytosol, that kind of keeps things cushioned and floating around. And that's what cytosol is. Chromosomes are also found in both of them. Those are our genetic material. And then ribosomes are something that make our proteins. So both prokaryotic and eukaryotic cells have a membrane, cytosol, chromosomes, and ribosomes. The main difference between eukaryotic and prokaryotic cells are that eukaryotic cells' DNA is contained in a nucleus-bound organelle, and it actually has a double membrane to it, 2 membranes. Whereas prokaryotic cells, it's just kind of like a mess inside there. It's all the chromosomes just kind of like spaghetti swirled around. So they have no true membrane-bound nucleus, and instead the gunk that is their DNA is called a nucleoid. They also have no membrane-bound organelle. So they're just super simplistic things. There's not a whole lot to study about these things. And that's kind of what it looks like. So if you were to crack it open, they don't have a true nucleus inside. They just have, I think it's on the right-hand picture, it's the kind of blue stuff. It's just kind of like spaghetti sitting around in there. And then they're much, much tinier. Eukaryotic cells are going to be a whole lot bigger than your prokaryotic cells are. So the plasma membrane, you kind of got a little bit of a feeling for what that plasma membrane was with chapter 3, I think it was, when we talked about the macro molecules, and we talked about lipids and how they had that like bilayer, and there was like a head that really liked to be close to water, but then the little tail hated to be around water, and that formed our plasma membrane. So our cells are surrounded by a membrane, just like our body is surrounded by the membrane being our skin. They're surrounded by a skin-like layer that is selectively permeable. I know you're going to have to know that term in lab. I can't remember when we really cover it for lecture, but I would know that it's a selectively permeable barrier, meaning that it gets to decide what it allows in and out. It doesn't let everything in, but it doesn't keep everything out either. So in essence, it decides what it wants to come inside. And it's made up of a double layer of phospholipids, one of those macromolecules. So it's a fat. It's a fat. So I can't zoom it out, but if you look at that image down there, the little gray balls are your hydrophilic, loving water heads, and then the tails are the hydrophobic regions that are afraid of water. And so the two scared parts of water are going to flip over and touch. And so it makes this bilayer where the inside and the outside, or outside and inside, are allow like to be around water, but the the middle of the layers don't like it at all. And you'll also notice that in that plasma membrane, we have other stuff that's embedded in it, like proteins, and there can be cholesterol is what that looks like on that those green little things. And some of those proteins go through the membrane and some of them sit on the outside. I want to say that we have a whole chapter where we're just going to talk about the plasma membrane. So we're just kind of skimming the surface on what the plasma membrane is right now. But know that it's made up of phospholipids. It's a bilayer, meaning it's two pieces, and the phospholipid has a hydrophilic head and a hydrophobic tail, and the hydrophobic regions will touch. This part is about cell size. So one of the reasons why when we grow, our cells just kind of bud and separate, and our body makes same size cells, just daughters of a mother cell, as opposed to a cell just getting bigger and bigger and bigger, is because a lot has to happen inside that membrane. There's a lot of metabolic activity that occurs there. So it is more efficient to have lots of tiny cells than it is less very, very large cells. If you have a very, very large cell, you might be able to cram more organelles inside there, but you actually have less surface area. So this is an image that is put in every single biology textbook, and when I took it, it didn't quite make a whole lot of sense to me. But the point is that if you have one little teeny tiny cube, you can see what the surface area is. If you expand that cube, you multiply it times 5 all the way around, your surface area is 150. But if you just were to take that little teeny tiny cube and pack a whole bunch of cubes all around in the same volume as that giant cube, you see how much more surface area you actually have. So cells have to be small because surface area, the plasma membrane, is essential to being able to do the work that the cell does. I'm just making sure that is what this slide is covering. Okay. So if you go there's this animation of the tour of the animal cell, and then there's also one that's a tour of the plant cell, and I don't think I'll play it for you guys because you can find it in Pearson under study material. I can't remember what the link is. I think it's called study material. I don't have my Pearson stuff up. And then you can choose your chapter, and you can actually choose videos and images and things like that. Do y'all kind of know how to get to the animations and stuff? Okay, so you know how to find it. I'm not going to play it for you, so y'all can get on with your Sunday. But I would, the biggest questions, or the majority of the questions that you're going to get on the exam, are going to be over what these organelles are and what their function is. So their names, their functions, and also a little bit about where they're located, meaning like a nucleolus is inside the nucleus, not like it's on the bottom right hand side of every cell, because it's always moving around inside there. So I would know all the organelles. I would definitely know their functions, and I would also know the difference between animal cells and your plant cells. So we're going to go through all of those little tiny organelles. The first being the nucleus, which is found in both the animal and the plant cells. Nucleus is where we have all of our genetic information inside of us. And also I would know that, so we have another organelle called a ribosome. I think we'll talk about those a little bit more in depth in a minute. But the general idea of what the ribosomes do is that they go into our nucleus, they take information from the DNA to make protein. So build muscles, make proteins that will be in your plasma membrane, all the proteins that are all over your body. So ribosomes make proteins, and they get the information to code the proteins from the DNA, from your genes. So all of our genetic material is inside of our nucleus, and it is the largest, and generally it is the largest. In plant cells, that's not always the case, but it's like the the most dense organelle. So if you were to take like the toothpick and look at your cells under a microscope, you can almost always find your nucleus in there because it's just really dense. And one of the reasons for that is because the DNA is really dense and it's all squiggly, and so it fills up this space and it gives it a good shadow. You don't really see much detail other than a little tiny blob inside of a little blob. And the nucleus is enclosed in something called the nuclear envelope. And the nuclear envelope is also a double membrane. It's a lipid bilayer, because lipid bilayers are super duper important in things that happen inside the cell. So this is what a nucleus, this is an animal cell, and the dark thing in the center is the nucleus. This cell has been stained so that it looks differently. You know, if you were just look at it in the microscope, you would not quite see it the same way. But it's a big glob region in the middle of your cell. There has to be things that leave the cell. So DNA will make RNA. RNA leaves the cell and it gives information to the ribosomes. And the way that things leave and enter your nucleus is through a nuclear pore. It's just a little hole that they go in and out of in that nuclear envelope or the nuclear membrane. And then nuclear lamina is a bunch of like straw, like really tiny string-like but tough. It's like filled with pipe cleaners. I guess that's the best way to explain it. So these are little proteins that look like pipe cleaners and give that nucleus its popped out full shape. You never really see a smushed down pancake nucleus. It's always really big and fat. And the reason why is because the inside is called that nuclear lamina. So I would know the envelope, the lamina, are all inside of the nucleus. This is just an image of that envelope. And then all of our genetic information, all that DNA, of course, is called our chromosomes. So they're organized into things called chromosomes, which are generally kind of like squiggly, spaghetti-like things. They will start to condense themselves before your cells will separate. We will go over that. I think it's chapter 9, maybe, that's mitosis and meiosis, and that will make more sense. Chromosomes. So chromosomes also have DNA in them. I'm sorry, they have DNA, but they also have proteins in them. And so the proteins, the DNA and the proteins of the chromosomes are called chromatin. So chromosome is just the genetic DNA material. When you add the proteins to it, it becomes the chromatin. And that chromatin is what condenses to make, you know, like you see images all the time of like two DNA molecules. When they're condensed like that, they're called chromatin. And then so the nucleus almost has its own nucleus. The nucleus has something called a nucleolus, and that's where RNA is synthesized or made. Synthesize means to make. And that specifically is where ribosomal RNA is. So it's RNA that's meant to go tell the ribosomes what information it has. It gets this code and it leaves the cell and it gives it to the ribosomes. The ribosomes then make the proteins. Okay, that's exactly what that is there. So we have two different types of ribosomes. Some of them are just floating around like crazy, and they're itty bitty little beads that are just floating around. But then we have some ribosomes that are attached to another organelle called the endoplasmic reticulum, which leads us, I think, to talk about the endomembrane system. So ribosomes make proteins. They're either independent, free, or they're bound, and they're bound to the endoplasmic reticulum, which is part of an endomembrane system. And the endomembrane system contains a lot of different organelles. So we already talked really about the nuclear envelope, but so it's a system that's made up of lots of organelles that have lots of folding membranes. They are membranous organelles. So nuclear envelope was one of those. It's what surrounds, it's the layer that surrounds the nucleus and keeps it having its shape. Then we have endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and plasma membrane. So we're going to talk about what each of these specifically do, but that's a good, if you're making note cards or something, that's a good thing to remember, is what makes up the endomembrane system. And then all these components, they can transfer information. They can talk to one another either by touching one another, or they have these little vesicles, which are like little tiny vehicles that will pop off of one and then go fuse with another in the endomembrane system, to share information with one another. Because it's like the cell is like a whole little town. It's like a little city, or factory going on. There's lots of information that has to be spread. It's amazing, like if your body can function as a whole, but then if you break it down, there's so much happening in our body all the time. Endoplasmic reticulum is part of the endomembrane system. For short, we call it the ER, and it has two different parts to it. We have smooth ER, which will have no ribosomes, and then rough ER, which is stuck with ribosomes all over it, and it looks like it's rough. It's bumpy in look, and that's why it gets the different names, smooth or rough. And it is so many folding different areas. So it contains most of the membrane inside of a cell. And this little video just kind of shows you, I think they're putting dye through it, so you can kind of see where all the folds and all these little different fingers are that make up this endomembrane system. It looks like a blob. And then I believe that the little dots there, so this must be rough ER, I believe that all the little dots there that are kind of traveling with that movement are the ribosomes on the rough ER. So the ER is kind of, it almost surrounds the nucleus. It's attached to it, and you can see there are parts of this ER that are smooth and that are rough. So it's not necessarily like you have a rough piece and then a separate organelle over here that's smooth. Within the same big giant organelle that's all connected together, you have rough and smooth portions of it. And I think it's kind of neat how you can see it on a electron microscope image, how you can see these right here. You can see the ribosomes actually attached to it. There's little tiny dots that are stuck to the parts of the ER. Close this. So the smooth ER and the rough ER have two different functions actually. The smooth ER makes fats, and then it metabolizes carbohydrates, so it breaks down carbs and makes fats. It also detoxifies drugs and poisons. So you would, a good thinking forward kind of question would be like, somebody who is addicted to drugs or has to take a lot of aspirin to be able to move around every single day, and they've noticed that they are having to take more and more and more of this medication to feel the same, you could assume that they have now developed a lot more ER in those cells because they're starting to detoxify those drugs. Or like your liver that's known to detoxify things would have a lot of smooth ER in its cells, as opposed to maybe your heart muscles. And then it also stores calcium ions. So it makes fats, breaks down carbs, detoxifies drugs and poisons, and stores calcium. The rough ER has the ribosomes on it, so it's really responsible for, it makes a lot of membranes, because membranes are made up of proteins, fats and proteins. But so if it has ribosomes associated with it, then it makes sense that it would make lots of membranes for us. So it distributes transport vesicles. Transport vesicles are proteins that can allow things to go through membranes. It secretes glycoproteins, which are, which really actually, that was what this green stuff was. That wasn't a carb, like I was saying. So those glycoproteins are those little green things that are kind of popping off of a protein. So they're producing all the things that we put inside, that our body puts inside of the plasma membrane. So smooth ER does a lot of stuff. Rough ER is really more is really focused on making proteins for our membranes. And that's why they call it the membrane factory. Our Golgi apparatus is like the UPS center of your cell. So the Golgi is a lot of also flattened membranous sacs. They call them cisternae. So you can see them here. It's not as like, it's like looping when it's the ER, but the Golgi is a little bit different. It has these little vesicles on the ends, or these little pods that you see popping off on the edges. And so the Golgi the ER makes the stuff, and the Golgi takes it and makes it into it refines it, I guess is the best way to say it. So it will make some macromolecules, it modifies and refines what the ER is making, and then it sorts, but its main thing is sorting and packaging materials into these little transport vesicles to go, and it tells it where to go and take that information. So that's why I say it's like the UPS or the FedEx. I guess you could say now it's like the Amazon. So it's taking stuff in these little, so it'll pod off right there, right? And then those things move off and go off somewhere into the cell. You can actually see some of these round little pods coming off of our Golgi in that electron microscope image. I think this is a video that shows you all those little vesicles flying off all over the place, coming to the Golgi, leaving the Golgi. So those vesicles might be coming from like the ER, rough and the smooth ER, and then they'll take it, refine it, and then spit it back out again. But because they're all part of the endomembrane system, they can talk to each other because the membranes in those vesicles communicate better with one another. That's just a 3D image. This is secretion, so it's going to be getting rid of stuff, sending information out. Kind of looks like shooting stars. Okay, that's enough of that. Then another part of the endomembrane system are our lysosomes. So we have to have, our cells produce waste, and we have to have a way for that waste to go away. It can't just stay inside of our cells. So our lysosomes are those that help break down things like that, things that are trash. So it's a super duper duper acidic environment. It's kind of like our stomach. And it has a very special cell or membrane around it so that it doesn't dissolve its own membrane, because it actually will engulf and take in trash vesicles and then dissolve it. So it has to have a special membrane itself so that it can dissolve everyone else's membrane but leave its intact, so it's not just squirting out acid all over the cell or lysing things.
I have a question about lysosomes.
Yes.
I read in the book that lysosomes break down mitochondria, but I was wondering if it was just mitochondria or all things that are no longer functioning.
It'll take all things that are trash, and usually it'll like like if the Golgi makes something and it's not perfect, then it will package it and send it to the lysosome, or the lysosome's just kind of wandering around figuring out what's trash and what doesn't belong. Or if something gets into the cell, like a virus or something that shouldn't be there, the lysosome's job is to find it and try to get it out of there. So let's see. So one way that when they engulf something else, it's a term called phagocytosis. When we talk about cell-to-cell relationships or interactions, we'll talk about phagocytosis, pinocytosis, and endocytosis. These are just ways that cells eat things. So phagocytosis or phagocytosis is where it actually turns into like two little hands, and it grabs it, fuses, and now that trash is inside of its stomach. It's like it has a mouth, and then it engulfs it. So I think this is an image. I don't know how long this is.
Like secretory proteins and some other proteins destined for lysosomes are made on ribosomes bound to the rough ER and move through the...
Can y'all hear that okay, or do I need to unshare and then change it to where you have computer audio?
I can't really hear it.
You can't? Okay. Let me I'm going to stop sharing and reshare it with computer sound. Okay.
Other proteins. Proteins destined for lysosomes are made on ribosomes bound to the rough ER and move through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Actually, that was a really stupid video because it was just kind of confusing. Here, this might show.
Phagocytosis is the ingestion of a solid, such as a microorganism or cellular debris, by a eukaryotic cell. Phagocytosis is used by some protozoans for obtaining nutrients, but this process is also used by certain cells of the immune system to fight infection. These immune system cells include macrophages and neutrophils and are collectively called phagocytes.
So the point of that, you saw those little fingers come out and suck it inside. That's what phagocytosis is. They reach out, they grab it, and they engulf the whatever the trash cell is in some way. And that's an image of it. Okay. Vacuoles are also part of the endoplasmic reticulum, but vacuoles usually, well, there's different types of vacuoles depending on if you're an animal or a plant cell. So food vacuoles are formed by phagocytosis. So they engulf other components. If cells have to eat themselves, so they're grabbing onto things. So protists are generally, they can be unicellular organisms, even though they have a eukaryotic cell, but I guess they can be multicellular too, maybe. But anyway, the protists, and maybe that's what that was trying to show. Protists will use contractile vacuoles to pump water out of their cells. So a vacuole is just kind of like a compartment that is used for food, for water, for structure too, because if you pump yourself full of water, then you're going to fill up, you're going to expand a little bit more. And then in plant cells have central vacuoles, which are really important for them for maintaining their shape. So a wilted plant has it has vacuoles that have shrunk down, and when you water it, the vacuole sucks up all that water and it becomes like robust again and strengthens itself back up again. Hold on, I've got like things popping up that I've got to make go away somehow. There, okay, gone. So if you look in a plant cell, the central vacuole is like the biggest thing inside of it when there's water in it. So it holds all the excess water, so that when it hasn't rained in a long time, but you're still able to see plants that don't look like they're wilted and dying just yet, it's probably because they have stored all of that water in that central vacuole just for moments like that. So vacuoles are just compartments that hold stuff, and I would know that central vacuoles are only found in plant cells. And then, okay, that's just a review for that. So again, know the components of the endoplasmic reticulum and what they do. That's the big stuff. You can make little cards for it. There should be like a quiz yourself thing where you can match things. And then I think on the quiz and the homework, I've done that where on the homework at least, try to do activities where you have to match the function with the organelles to give you that practice. So not part of the endoplasmic reticulum, we have organelles that help give our cells energy. And mitochondria are things that are found in animal cells, but we also know, I think we have mitochondria also sometimes in our plant cells. Chloroplasts are only found in plant and algae cells, so only plant things, only green stuff. So when I think chloroplast, I always think the color green, and in reality, they actually are green when you look at them. And then we have things called peroxisomes. These guys make water. So that's really important. Remember when we talked about like dehydration and hydration reactions, when you add water, you break things apart. So our body needs water to function, but also cellular, your body's using the water in a different way to make reactions happen. So the big thing about mitochondria is that they use oxygen and make something called ATP. ATP is cellular energy. It's food for your cells to make your cells do work. I don't remember what chapter it is, but we will talk all about mitochondria and chloroplasts, cellular respiration, and photosynthesis in those chapters. So there's this theory called the endosymbiont theory that arose as a way to help us understand and figure out how eukaryotic cells happened. So I like this slide a little bit better. So the endosymbiont theory is where they think that an early eukaryotic cell ate a prokaryotic cell, and then instead of digesting it, just formed a relationship with its host. It's kind of like, it's so simple that you think that it took scientists forever to, it's like it's made up. And then the host cell and the endosymbiont merged, so it maintained a relationship and they became one. And the eukaryotic cell had the mitochondrion. So then they think later on that at least one of those combined cells then ate a photosynthetic prokaryote, and that's how we ended up with chloroplasts. It's just really silly to me, I think. So know really briefly what the endosymbiont theory is. You might get one question on that. You're not going to get a whole lot of them. And it's that an early eukaryotic cell ate a prokaryotic cell, fused together and became one, and became what we understand eukaryotic cells to be. And that's how the eukaryotic cells probably got their mitochondria. I'm going to keep skipping. So mitochondria in this image are those orange colored deals. And inside the mitochondria, it looks like there's all these little squiggly lines. Well, it's actually the inside of its membrane. It's got a really wrapping, more surface area with all those little teeny tiny folds. And it needs a lot of surface area because that's how it does what we call cellular respiration. That's how it makes that thing called ATP. So the more, if you were to stretch it out, that inner membrane would be gigantic, but that means it allows in this tiny space for it to have all this room to produce more energy for your cells. So mitochondria is in nearly all eukaryotic cells. They have a smooth outer membrane, right? That wasn't very big, but the inner membrane had all those little folds, and those little folds are called cristae. The inside of it, in between the outer and the inner membrane, you have the inner membrane creates two compartments, the inner membrane space and the mitochondrial matrix. So you have the smooth, I wish I could get this to blow up. Let's do a little draw thing. I don't like yellow. So there's you can kind of see that there's a space between this really smooth outer membrane and where all the bends and stuff are. So this space in between here is called the inner membrane space. And then the part that has the dots inside there, those are ribosomes. That's your mitochondrial matrix. Matrix is like what is found within something that like kind of holds it together. So our blood is kind of our matrix. Blood cells make up the matrix inside us. Flour would make up the matrix of a cake, if that helps explain it a little bit better. So cristae are the folds. There's an inner membrane space. Then there's a mitochondrial matrix. And then some of the metabolic steps of cellular respiration are in the matrix, but most of it is actually across that inner membrane. Large surface area, the purpose of that is the bigger the surface area, the more ATP it can make. So mitochondria takes oxygen to make energy, so that's why it's found mainly in animal cells. Chloroplasts take light energy and convert it into food. into chemical energy or sugar. So we don't do that as animals. So chloroplasts are only found in animal cells. They capture light and turn it into energy in some way. And your chloroplasts are made up of different structures. So a whole chloroplast has, it's like if you make pancakes, that's your thylakoid. So you have these little pancake little things that pile up on top of each other. When all of your pancakes are stacked up, you have a short stack. So thylakoids are your pancakes, and when all your pancakes are stacked up, that's your granum. So in this picture, you can see you have lots of different thylakoids and lots of granum that make up a whole inside of an entire chloroplast. Chloroplasts have lots and lots of short stacks inside of them. The stroma is the inside fluid in there. So those little pancakes are not falling all over the place. They're being cushioned by the internal fluid called the stroma. The chloroplast is one group of plant organelles called plastids. That's just a term, but we don't really go much more into that until we talk about photosynthesis. So, when we talk about ATP and cellular respiration and we talk about photosynthesis, we'll look more at these organelles, but for this, we're just skimming the top of it. Mitochondria produces ATP. Know that what we see inside the mitochondria, it's got the folds or the cristae. The inner membrane is much, much more folded. The purpose of that is to give it more surface area. And then our chloroplasts have lots and lots of thylakoids that are stacked up. A stacking of thylakoids is called a granum. And there's lots of those inside of your chloroplast. And the fluid that's rolling around in there is called your stroma. Just like inside of our body, we don't have anything that's dry. There's always some fluid around your brain, in your body. Same thing with the cells. Something has to cushion that stuff. And then the last thing I think on organelles here are peroxisomes. Those are the ones that make water. So they produce hydrogen peroxide and then they convert that into water. So peroxide, peroxisome, that's an easy way, at least for me, to remember the two. And they're just kind of a conundrum. We don't know how they evolved or why they would have evolved, but a lot of them are found in the liver and they detoxify alcohol. So somebody who is an alcoholic would have a lot more peroxisomes than somebody who's never drinking a day in their life. Those are always fun questions to add inside the exam. So one thing I was going to say about this won't be on the test, but it's just kind of fun fact, useless trivia. Mitochondria, mitochondria, since it produces a lot of energy, is found in your muscles that need a lot of oxygen because it produces oxygen too. So when you have dark meat is like duck, right? And I guess it's goose too, but the legs of your chicken, not really chicken breasts because they don't really fly. So they don't need good, strong breasts. But one of the reasons that dark meat appears dark is because there's more mitochondria in it, which gives it that darker look to it. I don't know what the relation is with the greasiness, but dark meat is the dark color because more mitochondria is packed into those cells. So different cells, different parts of your body that do different things are going to have more of one organelle than others because they need more of it. So you don't have like identical numbers of mitochondria in every single cell across your body. I think one of the last sections here is the cytoskeleton. So cytoskeleton is so our cell has all these organelles, and I don't know if we mentioned this, but the matrix inside that's called the cytoplasm. It's like this. So when I was in high school, teachers would always make like a big blob of gelatin, and then we would smash in there different things that were supposed to show you what the organelles were. But the cytoskeleton is like a big gelatin, jello type thing. It helps give structure to the cells so that they don't smush down, and then all the organelles get smushed down. I'm going to skip through these things. There's the image of the cytoskeleton showing them. It's lit up, and you can see the nucleus right there in the center. So cytoskeleton is meant for support and helps it maintain its shape. It also is anchored, so some of the organelles can hang on to it, so they're not just floating all over the place. If they've got to stick still, then they're able to hang on. And then they'll interact with proteins called motor proteins to get movement happening. So some cells actually can move. And a really good example of that is, I thought there was a good video of it. Maybe not, maybe we'll come across it in a minute. So anyway, cytoskeleton interacts with motor proteins, allows cells to have mobility and actually move. Inside the cell vesicles and other organisms can use the motor protein feet to walk along the tracks provided by the cytoskeleton there. So organelles are able to, the organelles are also able to move because of the cytoskeleton, move within the cell. And the cytoskeleton is one of three things, or it is a combination of these three things: microtubules, microfilaments, and intermediate filaments. Microtubules are the fattest, microfilaments are the smallest, and intermediate is in the middle. And I just remember that filaments are, a filament is, a file is smaller than a tube of like toilet paper or a tube of paper towels. But they're all micro because they're itsy bitsy. I think this is just a video showing, it's just a terrible video, an old terrible video trying to show you the cytoskeleton. Here is actin crawling. Apparently crawling very slowly. Chloroplasts moving.
In these cells of Elodea, a water plant, the central space of each cell is occupied by a huge central vacuole, around which you can see cytoplasmic streaming. In the stream of cytosol, you can see mitochondria and also chloroplasts, the larger pigmented organelles. This movement allows nutrients and metabolic products to reach all parts of the plant cell. Credit Michael Clayton, University of by a huge...
That's pretty cool because, so you can actually collect these. We used to do this in lab, but then people couldn't figure out where to find a good pond that has a lot of algae and other just green, gross junk in it. But you can collect these little plant cells, and it's it's totally a vacuole because they're saturated and sitting in water. So they've absorbed all of this water, but those little dots that you see kind of floating around it are your organelles moving around and being able to move because they're able to grab onto the cytoskeleton. And so then this, I guess it's a table, not a figure inside of your text, shows you the structure and function of the cytoskeleton, the microtubules, the microfilaments, and the intermediate filaments. The thing that I really care for you to know more than anything is just that they are part of the cytoskeleton. And then microtubules are hollow rods. So they're like tubes. They're like toilet paper, the inside of a toilet paper or a toilet or wrapping paper tube. And they are the function. So kind of what they look like, and then their general function: shape and support and guides movement. The microtubules are also important when we do cell division. They help pull the chromosomes apart. So you don't end up like Down syndrome, one of the chromosomes didn't get pulled apart and you end up with 3 instead of just 2. So those microtubules are also very important in reproduction and division. And then centrosomes and centrioles are in animal cells. They are, I would just know that this is a type of microtubule that is used in animal cells for reproduction of the cells, for budding off of the cells. It's what kind of pulls the cells, the genetic material away from one another, separates your chromosomes. And that's kind of what they look like. You can tell they're microtubules because you can see the hollow tubes inside there. And these guys will, so since you have 2 of them, they'll move on either side of the cell. They call them poles. So they'll be on like the North Pole and the South Pole, so that the cell can pinch right down the middle and separate itself out. And so the older the cells are, the harder it is for those centrioles to do what they do. And so like That's why they think that when women hit a certain age, or as you age, you are more likely to have birth defects, because the centrioles are getting shorter and they're not as strong, and they're not able to do the proper division that should happen. That's why Down syndrome generally spikes when mothers more than fathers are older. So cilia and flagella are also microtubules that are on the outside of cells that will beat. So a cell that has a flagella would be like a sperm cell. It has a tail that allows it to move. And then cilia are basically like little hairs that just kind of beat and move and allow little organelles to move, usually little things that live in the water and stuff like that. And you can see that they are tubules because there are holes in there. So here's a good one I think this is a good one of ciliary motion. It's just the outside of something, but like little protists will have hairs. You usually see images with little hairs all over their bodies. Paramecium will have them, and that's how they get moving. And then these should be sperms. You know, everyone's seen look like.
See how a flagellum beats in the presence of ATP, even when separated from the cell. The flagellum of one of these bull sperm cells was cut in half using a glass microprobe. Notice how the excised piece of the flagellum continues to beat in the absence of any connection to the original cell.
Just because it has energy. I don't really care that you know why that was beating, but the point is just to give you an image of those microtubules moving and rubbing against each other are causing a little tail-like motion. I don't really care that you know what the basal body and the dynein are. So we won't even really talk about it. But the dynein, that's what helps it walk. It's just a protein. So tubules would be like cilia, flagella, and then centrioles for reproduction of the cell. Microfilaments are solid. They're smaller, and they're not a hollow tube like the tubes were. And their job is to bear tension, resist pulling forces. Microvilli of your intestinal cells are examples of microfilaments. I thought I had a video of it, but I don't. So they use a protein called myosin to get them to be moving around. And there's not a whole lot much more we're going to talk about them. There's a lot more you want to learn about microtubules than there are microfilaments. And then the intermediate filaments are somewhere in between in size, between the tubules and the filaments. So they're larger than the filaments, smaller than the tubules, of course. They are found in only some cells of animals. And they do the same thing really that tubules do. They reinforce cell shape, and they'll also fix organelles in place. So that is different than the tubules that allow things to move. These will actually, cram them. They get sticky. So it's like Spider-Man's web, making them stay stuck there. They're more permanent elements of the cytoskeleton than the other two are. Concept 4.7, section 4.7 is about the extracellular components, and then kind of how they talk to one another to coordinate different activities. So most cells synthesize and secrete materials that are external to the plasma membrane. I should have said this before I got started, but I uploaded smaller PDF versions of these notes into Canvas. It's under the PowerPoint presentations page or module, so you can click on that, and if you want to download and then print these off, they're available there. Should have said that in the beginning so y'all could be taking notes off that. I apologize. So our plants have something called a cell wall that is not found in animal cells. So one big difference between animal cells and plant cells is that plants have a cell wall and animals do not have that. And the cell wall is what gives the plant cells their rigidity, and that's why they can maintain their shape for the most part, even when they don't have a whole lot of water, and even when the vacuoles start to shrink in. Instead, they have a cell wall, a strong, rigid cell wall, and inside of that a cell membrane, which is what our animal cells only have. And so the membrane will shrink in, but the cell wall stays relatively firm. That's why even though the plant gets wilted, it's still it doesn't just doesn't just shrink up into nothing. It's not like, when spinach sits out too long or something, it doesn't do that right away. So the cell wall is made up of different layers. There's a primary cell wall, a middle lamella, and a secondary cell wall. Primary cell wall is pretty thin and flexible. The middle lamella is in between, and the secondary cell walls is not in every single plant cell, but it's in some of them, and it's between the membrane and the primary cell wall. So if you pop it up here, I guess what they're trying to show is that maybe kind of the outline of the whitish beigeish stuff is going to be your cell membrane, and then the cell wall is that orange peachy color, and then the gray, right? So primary cell wall is on the outside, and it is the peachy orange color. Secondary cell wall is thicker and more rigid. The primary was thin, and that's the brown stuff. And the middle lamella is the space in between two plant cells butted up next to one another between their two primary cell walls. Isn't always necessarily there, not in every single cell. So animal cells do not have cell walls, but we have that we have a very elaborate extracellular matrix on the outside of our plasma membrane, on the outside of the outer layer of the cell. And I think we'll show you some I'll show you some pictures of that, but if you remember back to that, one of the very first figures that we were looking at, there were proteins embedded in there. There were things popping out all over the place. There's a whole lot happening on the outside of the cell that interacts with other cells, gives it rigidity, gives it purpose, makes it do different things. So we might have on the outside of our cells glycoproteins, collagen, proteoglycans, fibronectin, but also when you saw that protein that was going through the cell, those are called integrins or integral proteins because they integrate through the plasma membrane. And then you lose your collagen as you age, and that's how wrinkles and things like that happen. So that there's the extracellular matrix right there. So where it's kind of brownish down here, that's the inside of the cell. The blue stuff on the outside, that's the outside of the cell. There's fluid around the outside of the cell. You have you have collagen, you have different proteins sticking up, you have all these different things, microtubules and other stuff that are on the outside, giving it some structure, helping it communicate and talk to things that are other cells that are around it. And we have different spaces between our two animal cells, between our neighboring cells, animals or plants actually. And they talk to one another because they might say, I want some of you have more ribosomes, can you share it? Or, they might pass on genetic information between the two. And there's different types of communication or junctions when they neighbor each other: plasmodesmata, tight desosomes, and gap junctions. Plasmodesmata are just channels. And these are in plant cells. There's just perforated channels where information goes through. So you can see it in this electron microscope pic. You just have these little areas, these little tubes. So this is cell number 1, and this is cell number 2, two separate cells, but they're neighboring cells. They're nearly touching one another, and they form these, basically their cell walls have kind of reached out and fused and formed a little tube so that they can pass information between one another. So that's plasmodesmata, spaces between that make a little tube. Tight, and that's in animal cells. Tight junctions, desmosomes, and gap junctions are the common ones in animal cells, especially in epithelial tissue. That's where a lot of communication happens. I don't want to show you all that until we really talk about it, but is it going to show up? No, we're getting to the end of it. So here's a video of, I think it's of all of them, even though it says desmosomes.
Animal cells and multicellular tissues are usually joined by tight junctions. Desmosomes, also called anchoring junctions, and gap junctions, also called communicating junctions. Plant cells are often joined by plasmodesmata. Each type of junction has specialized functions, such as preventing fluid leakage between membranes or allowing the free passage of small molecules and ions between adjacent cells, found predominantly in epithelial. Tight junctions, which bind cells together, are found predominantly in epithelial tissues, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells. Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins, such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton. In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow the passage of water and small molecules. Gap junctions help coordinate the activities of adjacent cells. For example, a hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions. Plant cells, unlike animal cells, are surrounded by thick walls that form rigid tissues. They do not need the junctions found in animal tissues. But some higher plant cells are interconnected by plasmodesmata, tube-like structures that penetrate through cell walls and form fluid connections between adjacent cells. Like gap junctions in animal cells, plasmodesmata allow the free exchange of small molecules and help coordinate the activities of neighboring cells.
That was actually a really good animation if y'all want to. I guess it was called Desmosomes. If y'all want to watch that one later on, because it explained all 4 of the different type of junctions between cells, animals and plants. And then I think the last comment, or maybe even be section 8 in that chapter, is that the cell is greater than the sum of its parts. So while all of those organelles are really important and very interesting, what they all do together is more productive than what they can do on their own. Just like a team is greater than each individual player. So that's, we spent a whole hour just on chapter 4. So the majority of the exam questions are going to be on chapter 4, but there is a little bit on chapter 17, a very little bit. We're only covering section 1 and 2. And we never know exactly where to stick this chapter in our in the schedule for Bio 1406. We used to have it a little bit later on. We started moving it a little bit closer right after cells. I don't think it fits well here, but it honestly doesn't fit well anywhere else. So it's not my favorite chapter at all, because it's almost like it doesn't need to be covered. And there are certain concepts in this chapter that we are nowhere close to really talking about. And that's one reason why I don't like it so much, because it's very confusing. So when I test you over it, I try to just keep it very, very like skimming the surface, really not getting too deep on anything. So basically, know what a virus is. A virus is a parasite. It cannot live without a host. So it's an infectious thing that really doesn't have a whole lot to it. It's a very, very simple kind of an organism. It really it doesn't have like a nucleus or anything like that. It's just a really simple little thing. Consists of little more than its genes that are packaged into a protein coat. I don't know what that is a picture of. So we really can't see viruses, even the very biggest ones. You can't see it with a light microscope. So you have to look at them with an electron microscope. Viruses are not cells, so they're not their own cell. They're a virus. and they are they're not even a prokaryotic cell. So viruses are their own things. They cannot reproduce without a host. So technically they're, where do they fall? Like in the grand scheme of living things, it's really hard to know where to stick them. They're not cells, but they're enclosed by a but they are a nucleic acid enclosed in a protein coat. Viral genomes, so they either have, they're weird because like all animals have DNA and we have RNA. Well, viruses, the genetic material of viruses is either double or single DNA, or it's double or single-stranded RNA. We only have single-stranded RNA in our body. So it just depends on the type of virus that it is. And oftentimes we'll call it, if it has DNA or RNA, we'll call it a DNA virus or an RNA virus. A capsid is this protein shell that covers the viral genome. So here are a bunch of different structures of viruses. They come in all different shapes. Some of them, like that one on the far right, looks like, I don't know, like something like something that astronauts would place on top of outer space on some planet. But they are all wrapped up in that viral envelope. So capsid is a protein shell that encloses the viral envelope. Capsids are built from things called capsomeres, and they have a variety of shapes. So like the capsomeres on this one that's shaped like a tube, the sum of the capsomeres, it makes up the capsid. This one looks like a hexagon, like a 3D hexagon thing. They come in a variety of different shapes. Capsomeres make up the capsid. That's what encompasses their DNA, but it's not technically a membrane or anything like that. Some viruses have membranous envelopes that help them infect their hosts. They're called viral envelopes. So 17.2C is an example of it having this envelope around it. It's not really a membrane, but they've got this little envelope. And the envelopes are derived from the host cell's membrane. So it's stealing the host, it's making the host cell's endoplasmic reticulum or make membranes for it that it's going to use. Viruses are just aliens. Bacteriophages are viruses that infect bacteria. So you might have heard the term phage before, and they're just a well understood type of virus. So we're going to look at, in this next section already, we're going to look at how they replicate in the host. And one way that we're going to look at is how they replicate in phages. So I would just know that a phage or bacteriophage is a virus that infects bacteria, which sounds funny. A virus can infect a bacteria, but they can. They have the most complex capsids. This last one that was really weird. It looks like it docks down and then it's going to inject its DNA material into the host cell. So they have a protein tail that injects into the DNA into the host. So viruses are called obligate intracellular parasites. Obligate because they depend, they're obligated. They cannot survive without the aid of their host cell. They can't survive out on its own. They're inside of the cell, so they're intracellular parasites. They can only replicate when they're inside of the host. So I would know what an obligate intracellular parasite means. The host range can vary. So some things can have a really narrow host range, like I don't know, like West Nile can infect birds and humans and horses. So it's kind of a medium host range, whereas some viruses only infect humans. So it ranges, and host range is the number of hosts that they can infect. And sometimes some viral infections can be limited to certain tissues. So you could have like a viral like West Nile is considered a encephalitis, so it affects nervous system tissue. You have other viruses that might affect other tissues. I don't know what coronavirus tissue affects. I'm assuming probably respiratory tissue, as we're learning, probably more than other tissues. So you want to know just really kind of generically how viruses replicate, the replicative cycle. So once the virus gets its genetic material into the cell, it takes over the cell and makes that host cell make more viral proteins. And then the virus, once it's all replicated inside the cell, will exit the cell. And sometimes it will damage or kill it or destroy it. Other times the cell continues to live. But it enters the cell, makes it replicate, and then leaves to go on, and it makes more of itself to go on and do more to other cells. I think this is a fairly good animation.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that's derived from the membrane of the host cell. There's usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell, and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA messages into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Okay, so that's actually a really good animation of just the general way that the virus infects the cell and then replicates itself. So enters the cell, makes this host cell make its own DNA. Then it also makes the RNA. Remember, RNA makes proteins. It makes it make more of those capsid proteins to encapsulate the DNA, and then it pops out and it moves on. So the replicative cycle in phages, which are bacteria that will infect, which are viruses that infect bacteria, is the best, most understood one. And so they have lytic and they have lysogenic phases. So a lytic phase is where they actually kill the cell. They culminate at the end, it ends in death of the host cell. Lysogenic, it will not do that. So lytic cell produces new phages, new viruses, and then lyses or breaks open the cell wall and releases all of those baby viruses. A phage that reproduces only through the lytic stage is called a virulent phage. So I would definitely know what that term is, that they reproduce only through the lytic stage. They only end in killing that, only end in killing that cell.
Bacteriophages, or phages, are viruses that... Bacteriophages, or phages, are viruses that infect bacteria. This is a T4 phage, which consists of DNA inside a protein coat. The lytic cycle begins when the tail fibers of the phage stick to receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes transcribe the phage genes and translate them into phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the lytic cycle of bacteriophage T4.
So one reason why I hate this chapter is because we haven't talked about, what transcription and translation and all that stuff means. So we're throwing these terms at you that are confusing. So that's why I try to keep it as simple. The questions that I choose, I think are pretty easy. The ones that I write, I try to make them just very, skimming the surface type of a thing. So lytic phase, it kills it. Lysogenic phase, it will replicate without destroying the host. So some phages can use both lytic and lysogenic cycles, and they just decide which one they want to use at that given moment. And so those are called temperate phages. So they are, trying to think of how I remember that. For some reason, temperate to me makes me think of like, not temperature, but like your temperament, right? So you never know what their temperament's going to be, if they're going to kill you or if they're going to keep you alive. So if they use both when they decide, it's called a temperament phage. So they have the choice on this left-hand side to do lytic, and then on the right-hand side, they're doing lysogenic, where they've entered the cell. They will actually integrate their own DNA into the bacteria. Then they split off, they bud off. So now that it was one bacteria, now it's two. Both of these have the virus's DNA in it. And so every offspring of that bacteria contains that virus. So it spreads and spreads and spreads. A viral DNA molecule is incorporated into the cell's chromosome. So the virus enters the cell, it inserts its DNA into the bacteria's DNA. When it's integrated, that's called a prophage. So that is a term to remember. Prophage is when it's a bacteriophage replicative cycle, when the virus is replicating inside of a bacteria only, and the virus has integrated its DNA into its host bacteria cell. That's called a prophage. And then every time the host divides, every daughter it has contains that virus. So one cell can actually give rise to a larger population in the end than when they explode the cell through that lytic cycle. We can watch that because it's pretty good.
Some viruses can carry out either a lysogenic or a lytic cycle. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. In the lysogenic cycle, the viral DNA is incorporated into the host cell DNA, where it's called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own DNA and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages, which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda. In contrast to the lytic cycle, the lysogenic cycle replicates the viral genetic material without destroying the host.
Okay. So you would think that there wouldn't be bacteria anymore. If you have these viruses which are detrimental to the bacteria cells or any cell that it's in, that if bacteria are single cellular organisms, then why on earth do we still have bacteria? Well, they have some defenses against these phages. So I would know the different defenses they have. One is that they have these restriction enzymes where they can find that phage and they can chop up their DNA, and so they can't get into the bacteria's DNA. They also have, whoops, did it move? Oh, it did. They also, okay, I'm confusing myself. They also have, there's also this thing called a CRISPR, CRISPR-Cas system, which are these little proteins that when the phage has integrated, well, I don't know. So this is one reason why I hate this chapter, because we're going to be talking about like RNA and how it, it has a prime and another end. Just know that there's a system called a CRISPR system, which is one of the defenses that bacteria have. How about that? We'll keep it an easy question on there. Well, I guess we need a little bit more about it. So when the phage infects the bacteria and it has the CRISPR system, not every one does. If the cell survives, if the bacteria survives, it now has this CRISPR system in its DNA, and it will remember if it survives and gets reinfected again, that CRISPR is like a essentially it's like a immune system for the bacteria, and it will go and find the DNA of that virus, and it will chop it all up. Also, it will ruin it. So the bacteria cell and its offspring are further protected if they have that CRISPR system in them, and not every bacteria has it. I know it's confusing. Let's see, we only have a few more slides. So that was how things replicate in bacteria. Now looking at how things replicate in animal viruses, there are 2 key ways that they do it. They're either a viral genome, or they have or do not have an envelope. If they have an envelope, the virus is able to use it to enter the host cell a little bit easier. And they do so by basically fusing and entering the host cell. Same kind of concept as we saw with the generic version, where their virus RNA makes DNA, so the virus makes its own genetic material inside the cell, forces our cells to do that for it. And then it will go and fuse. It also makes our ribosomes make those capsids. So it will remake itself, fuse with our cell, and pop itself out. So it uses its envelope because it has all the capsids to do that. The viral envelope is derived from the host cell's plasma membrane, so it uses the cell to make its own plasma, to make its own envelope. Most molecules of this membrane are specified by the viral gene. The gene tells it what to do specifically. And then some viruses have actually their envelopes have derived from the membrane of the Golgi apparatus in the host. So herpes virus is a really good example of this. And when it does that, it actually leaves behind some of its DNA in the Golgi and in that cell. And then every time your cell reproduces, it still has that virus DNA in it. And so like with herpes, you hear about when people have sexually transmitted herpes, or you have the herpes virus as like cold sores, you always have it latent in your body, and some environmental stressor, whatever kind of stressor, emotional stressor that you might have, is what triggers that virus to then become active again, and you have an outbreak. So yeah, that's what that is. That's why herpes comes back, is never totally gone, because it's that virus is actually integrated into the DNA of your body. The broadest variety of RNA genomes is found in viruses that infect animals. So retroviruses use something called reverse transcriptase, which is nothing we're going to talk about until we talk about genetics a little bit later on. So you just have to kind of know that a retrovirus uses this thing that we don't know about called a reverse transcriptase to copy their RNA and make it into DNA. So they convert RNA into DNA, but they do it, it's called a reverse. They do it opposite. So it's kind of confusing. That's all you need to know. They make RNA into DNA in the opposite direction of what we know is normal. And HIV is a retrovirus. That's what HIV is. So after it enters the cell, after the virus enters the cell, and it makes its DNA, makes the cell make its DNA, it integrates itself into the host's genome. And so when it does that, it's called a provirus, and it now becomes a permanent resident of the host cell. And every time that host cell replicates, it's still there. So you hear that you cannot get rid of HIV, right? It's something that you always have. And that's why, because it's integrated into your DNA and it's always going to be there. You can suppress symptoms, you can treat symptoms, but the virus is always going to be there. I'm not sure if I like this one or not, but we'll get started.
Retrovirus, HIV reproductive cycle. Understanding the structure and life cycle of HIV, human immunodeficiency virus, or the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of 2 molecules of single-stranded RNA. The enzyme, reverse transcriptase, enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named as retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane, and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
Lots of terminology that you don't know yet, and we're not going to learn for a while. So that's kind of the end of that. I guess this last point is how we think that viruses evolved. So the general theory among scientists is that there was a cell that broke, and then there were naked bits of just nucleic acid. So it was naked, meaning it wasn't bound by a nucleus, and that stuff may have been what a virus evolved from. But really, nobody knows. They're just weird organisms. So again, much more on chapter 4 than on chapter 17. If you have 40 questions on an exam, I would say you might have 10 questions of virus, and the rest would be on chapter 4. So, remember that as you're studying it. You put most of your effort into chapter 4 is what I would say. Do y'all have any questions before I log off from here?
No, ma'am. Thank you.
You're welcome, you guys.